The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 38 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, yeast makes an important drug from a plant root. Are we gender neutral when we pick people to referee papers? And why worms want to kill off males. But first, Staphylococcus aureus is a very common cause of infection in humans. In its drug-resistant form, MRSA, it can also be lethal. And one of the reasons that staph is such a challenge is because once it gains a toehold inside the body, it can be very hard to get rid of. For instance, if your hip replacement gets infected, often the only remedy is another trip to the operating theatre to remove the entire prosthesis. This happens because bugs like staph can assemble protective barriers around themselves called biofilms, which shield the bugs inside from the immune system and also fend off antibiotic agents. Now, two scientists at Rutgers University, Jeff Boyd and Amaya Mashawala, have discovered the trigger that makes Staph aureus begin building its biofilm. Jeff first.
If you've ever, let's say, walked in a stream and you've slipped on a real slippery rock, a lot of that slippery surface is actually created by microbes that are in biofilms, or these are these organisms that are living and have attached themselves to this surface. And it allows the water to then flow over these microbes, and they can kind of acquire the nutrients that are naturally present in the water, but yet they don't have to float and fight the current. Under a microscope, it'd be a cluster of grapes with maybe some type of a matrix or some type of a goo or a slime that might kind of hold those cells together.
And what was the question about biofilms that you were looking at here?
Well, it was kind of a serendipitous discovery, but really what we're interested in is how organisms make a shift between aerobic lifestyles and anaerobic lifestyles, meaning in the presence of oxygen or living in the absence of oxygen. And what Amaya discovered is just that when these organisms that we study grow in the absence of oxygen, they form very robust biofilms. And then we tried to extrapolate that as to what's going on with the physiology of this organism or why it might be doing or having this behavior.
And Amaya, what did you actually do to study this?
I looked to see how the specific bacterium called as Staphylococcus aureus, typically it lives on the human body and it doesn't cause us infection. But under certain conditions, it can cause infection. And people have found that its ability to cause infection is related to its ability to form these complex communities called as biofilms. So what I looked at is how Staph aureus stick to a surface and form these multicellular communities called as biofilms.
So how do they go from being an individual free-living bacterium to wanting, in inverted commas, to form this community coated by this biofilm, and what is it made of?
It turns out when you remove oxygen, it makes it want to go from living as an individual cell to start to living as a community. They start to cooperate. A part of this cooperation is that they start producing a sticky glue-like material called as the matrix. And this matrix helps these bacteria stick to each other, or it helps them stick to surfaces such as like human tissues. What happens is this sticky glue-like material, the matrix that the bacteria make, it kind of encases the bacteria almost like a raincoat. This protects the bacteria from components of the human immune system, so they can't come in contact with the bacteria and kill it. And this allows the bacteria to cause infections.
And Jeff, what do they make that bacterial raincoat from?
Well, I think this was one of the most interesting findings that Amaya made, is that it turns out that Staphylococcus aureus in these conditions is actually making this matrix or this glue out of DNA. When you remove oxygen, a certain portion of the population, these cells are killing themselves. And as they do that, they break open and they release DNA. And this DNA, it turns out, is a very sticky material.
And Amaya, do you understand how they get hold of the DNA, the surviving cells, and then use it?
That part is not entirely clear yet. However, what is fairly evident is that it does help them stick together in some manner. One idea is that in addition to DNA, The matrix is also made up of proteins. And DNA is negatively charged, and a lot of the proteins are positively charged. It's possible that they help kind of form like an electrostatic net, which then helps the cells, the proteins, and the DNA all come together and make this matrix.
And Jeff, we've heard that the bacteria kill themselves, but is that a choice, or is it that some randomly die and then their corpses get assimilated by the survivors? Or is there actually a predestined, you're going to die and this is how we're going to kill you, by the rest of the population?
I think that's a huge, outstanding question. We've found systems that are actually able to respond to oxygen. We found that certain cells break open, and we found that DNA is released. Now, how certain cells are chosen to have this behavior is still a huge mystery to us. We're not exactly sure what the stimuli might be that say, okay, you cell break open and you cell kind of form the biofilm and survive and take advantage of this lysing cell. But that is going to be one of the questions that we were going to try to address as we move this project forward.
And is it just bacterial DNA they'll assimilate? Because obviously one of the mechanisms, one of the virulence factors of organisms gram-positives like Staph aureus is that they lyse our cells and release various micronutrients, iron and so on, that they can then use. But inevitably there will be DNA coming out in the process. So could they use that too?
You know, it's an excellent question, and I'm going to have to say yes. Other researchers in the field have found that Staph aureus does produce a series of proteins that have the ability to bind DNA. And that DNA that they bind is not specific to the DNA of the organism that's producing these proteins. So I guess I would answer the question by saying yes, Staph aureus could take advantage of human DNA after it's lysed those cells to help facilitate the formation of these multicellular communities.
And Ameya, lastly, if you can get a handle on how this works and you can disrupt it, does this mean we potentially have a new way to combat Staph aureus?
Definitely, that's our hope. In a forthcoming project that we are looking at, we're trying to understand how we can take different molecules and make these biofilms dissolve and let antibiotics treat the infection. So our hope is that by understanding how this biofilm structure occurs, we should then be able to try and figure out a way to treat these infections.
Jeff?
Yes, I would agree with everything that Ameya said. One thing that he didn't point out, though, is that Ameya actually discovered the protein that has the ability to sense the presence or absence of oxygen. Now, knowing the signal for that regulatory system, we might be able to go through, work with some chemists, design some drugs to inhibit this protein that senses either the presence or the absence of this bacterium breathing.
Jeff Boyd, and before him, Ameya Mashruwala. A significant proportion of the drugs in the average doctor's medical bag have their direct roots in nature. Digoxin, artemisinin, morphine, and the salicylates are all very good examples. Another is forskolin. This is an incredibly useful therapeutic chemical, as we'll hear in a minute, but it's also an almost impossibly difficult molecule to make. So doctors have been forced to rely so far on a natural source, which limits supply, and it also drives up costs. Now, Irini Pataraki has found a way to make yeast brew it up for her.
Phoscolin is a compound that is produced only from one plant. The plant is called Coleus phoscolin, mainly grown in India and South Asia. And phoscolin is important because it has a lot of pharmaceutical properties that all of them depend on the ability that phoscolin has to help relaxing the vessels of our body. For example, phoscolin helps treating hypertension, asthma, glaucoma in the eyes. Actually is one of the very few drugs that they are efficient in treating glaucoma. And today we have in the market medicines that they contain phoscolin for that purposes.
But because it comes from one single plant source, we're therefore dependent on that plant to extract the precursors and the chemical.
Exactly. Also, we have to have in mind this plant produces phoscolin in relatively low amounts. So when we need phoscolin to give it to the market, then this source is not enough. And also phoscolin in that plant is produced in combination with many different metabolites. So this plant is producing something like 70 different metabolites, but quite similar with phoscolin. So it's very challenging to isolate and purify phoscolin from this plant.
And why can't we just make forskolin artificially, just in a test tube?
Because phoscolin is a quite complicated molecule, so it's very challenging chemically to produce it. And also it will be very expensive. The amounts that you can achieve are very low, so the price and the efficiency is not really favorable.
On the other hand, if you can work out how the plant is doing it and then steal the genetic recipe, then you could recreate the synthetic pathway in another organism and mass produce it.
And this is exactly what we are doing. We went back to the plant, sequenced the transcriptome, which means we sequenced genes that they were expressed in the plant, and we found out the genes and enzymes responsible for the production of phoscolin.
Did you focus on the part of the plant which makes the highest concentrations of phoscolin so that you knew you had a reasonable chance of the genes which are switched on there are the ones which are directly linked to making those chemicals?
Exactly. This is what exactly we did. So we knew that phoscolin generally is produced in the roots. And we knew that from the tradition because, as I said, phoscolin is used for many years. So people knew that they can find it in the roots. But when we started to work on that and we went closer, we found out that the phoscolin is not generally produced in the whole root, but is produced very specifically in the cork of the root. And then we found out that these cork cells, they contain some oil bodies where these oil bodies, they are able to store phoscolin and also phoscolin precursors. So but from this data, we realized that phoscolin is also produced in these cells, not only accumulating, but also produced. So we went back and we sequenced this very, very specific tissue. And this was a big help for the identification of these enzymes.
And how many enzymes are involved in the whole pathway?
We found 9 different enzymes that they are necessary to produce this compound. The difficult part, though, is to make all these genes to express together and to work together in a different organism and to produce what you want them to produce. Because the environment of a plant is very different than the environment of a yeast cell. A plant cell is much more complicated than a yeast cell, so you have to make some adaptations and you have to be sure that all these enzymes that you move from the plant to the yeast cell, you can make them functional.
Does the yeast tolerate making phoscolin okay, or is it toxic?
You could say that it's toxic, but because it's toxic, yeast has some nice machinery that excrete phoscolin out of the cell to the media.
And how much phoscolin does it make?
In this paper, we have said that it can produce up to 40 milligrams of phoscolin per liter of yeast culture. Actually, today we have managed to get a much higher titers.
And how does that compare then in terms of the amount of plant matter you would need to extract the same quantity of the drug? I mean, is this a viable and scalable way of producing phoscolin?
This is totally scalable because, of course, you can use as many yeast culture as you like. So you can have several bioreactors. While for the plant needs a whole year to grow, and then you have to chop it off and to take the roots. So when you rely on plants to produce phoscolin, you have to depend on yearly, let's say, growth.
An amazing piece of work. That was Irini Patareki from the University of Copenhagen. You're listening to the eLife podcast with me, Chris Smith. Still to come, how worms get rid of males they don't want, and injecting electricity into a human brain to boost cognition. Does it work? Before that, though, when a scientific paper is submitted for publication, an editor at the journal will usually select a number of authority figures in the relevant field and ask them to peer review the manuscript. So who gets picked for the job? Specifically, are male scientists more likely to be asked compared with women? Marcus Helmer.
We were looking into if there is any kind of bias in the way that editors or reviewers are chosen. In our specific case, we were looking into if gender matters for how reviewers are chosen. We used one specific series of journals, the Frontiers series of journals. They do publish alongside each paper who was the reviewer of that paper and who was the editor of that paper. They have been existing since 2007, and over the years they have published around 40,000 articles. these 40,000 articles, they were handled by 9,000 editors and around 43,000 reviewers. So altogether, that gives a huge data set in which we can study the behavior in the peer review process and look for potential bias.
And how did you interrogate that data set? What questions did you ask of it?
We were interested in if women are underrepresented beyond expectation. It's a well-known fact that there are just less women than men in science. If there are less women than expected, given that just numerical underrepresentation, and that seems to be the case. We saw that for authors and reviewers, while for the editors, the actual number was within the expectation.
Right. And what was the other question you asked?
The other questions we asked was if there's any bias in the way editors select the reviewers. And what we found is that it seemed that editors of both genders preferentially chose reviewers of their same gender.
Gosh, that's interesting, isn't it? So we have got a bias, but we've got both people being biased in favour of their own sex.
That's true.
And by how much?
It appeared that this bias is present for both editor genders, but it appears also that there's a difference in the way female editors prefer their own gender than in the way male editors prefer their own gender. It seems that this same gender preference is much more widespread among male editors, whereas for female editors it appears to be restricted to relatively few who are then highly preferential.
Why do you think we're seeing this? Because this is what we dub homophily, isn't it? Preference for your own gender.
Yes, so it appears that this same gender preference is something that might be just human nature in a sense. So you see that in small children already, for example, the way small children form friendship networks in work environments. It really seems to be a widespread pattern of human behavior.
Were you surprised by what you found?
We were surprised by what we found, yeah. We did expect to find that women are underrepresented, but that on top of that underrepresentation, there is also the same gender preference. We did not expect that. This is potentially also important because it seems that the number of women in science increases over the years, very slowly, but it does increase. And in a couple of years, even when numerical equity might be reached between the genders, the same gender preference might still be there. So by just improving the numerical ratio between men and women, the gender bias as a whole will not go away.
In other words, the system is a bit broken and people's approach to the system is wrong. And if we top up the system with women to achieve parity, it will still sort of relapse to type if we leave it alone anyway.
It appears so, yeah. So this same gender preference would have to be tackled on top of the numerical underrepresentation.
So nothing's ever simple, is it? That was Marcus Helmer, and he was talking to me from Yale in the US. Some species exist as hermaphrodites, and that means that a single organism can be both functionally male and female at the same time. In the case of the nematode worm C. elegans, though, it's also useful from time to time to be able to produce offspring that are exclusively male. This is because they can mate with other hermaphrodites to boost genetic diversity. But then you don't want too many males, because when they mate, they shorten the lifespan of their partner. And you don't want them hanging around competing for resources for longer than they're needed. So how do you get rid of them? Colleen Murphy has discovered the answer. They kill themselves off with a pheromone to which only the males are sensitive.
The species have what are called hermaphrodites. So the mothers make both sperm and oocytes. So they don't need males. They only use males in times of stress when, for example, there are conditions where they might want to increase their genetic diversity by crossing. So they want to produce a lot of males in those cases. And then when those males mate with the mothers, then 50% of their progeny will be male and 50% will be female. So you have these basically bursts of male production. And the hermaphrodites turn out to want to get rid of those males. We discovered they have a very clever way of doing that, which is the males themselves produce a pheromone that is actually toxic to themselves.
So you get this boom and bust population situation. There's genetic and there is environmental pressure to increase the number of males when you want to drive diversity, but at the same time you don't want that running out of control. So you hardwire into the system a death program so that as the density of males go up, they're secreting this pheromone that kills other males, so they will bring about their own demise.
Exactly. It's a very clever way, I think, of the hermaphrodites being able to say, well, we have these males, they need to be around for a while, but they're actually going to kill themselves by making this dose-dependent toxic compound. And that will eventually drive the population of the males back down so it resumes a mostly hermaphroditic population.
How do the males detect that pheromone in the environment then?
We assume it's through receptors, although the receptor for that male pheromone I don't believe is known yet. We certainly don't know it. And we don't know which neurons detect it, but we know that neurons are involved in this. And there's a genetic trick where you can make the neurons of those hermaphrodites males. When we did that experiment, those hermaphrodites both produced the male pheromone and they received male pheromone as a toxin.
Now, when a male mates with a hermaphrodite, that also has a negative effect on the hermaphrodite. Is it bypassing the neurological detection? Is it the same death signal, but it's just getting in via a different route? Or is that a totally independent mechanism?
That seems to be an independent mechanism. We've been able to find that those are due to the transfer of sperm and seminal fluid just by using genetic tricks where the males don't make either of those, but they still make pheromone. And the pheromone in that case doesn't affect the female. It's really the presence of this germline ramping up of activity from the sperm and unidentified components in the seminal fluid that make the hermaphrodite die early.
Have you contemplated doing the experiment where you just stop them being able to kill off these males like this and then just grow generations of them and see what happens to population fitness, population numbers and so on?
Yeah, so we've dreamed about a lot of these types of experiments. In our hands, it's been very difficult. These animals have at least 300 progeny each, and they have about 1,000 if they mate, and they quickly grow out of control. So there are lots of thought experiments that we've mulled over, but we haven't actually carried out any that are multi-generational, at least not successfully. We've done a lot of modeling to try to figure out what would happen if you just, for example, inhibit their ability to be fertile or produce progeny, and that's how we were able to show that prediction would be that they'd go back to a mostly hermaphroditic species after several generations. Certainly losing it, we haven't thought so much about that. We're really mostly interested in trying to figure out the mechanism that's at work there now. Like, what is the mechanism by which they actually kill the animals? Because that's something, it doesn't look like any of the other lifespan or longevity pathways we've worked on previously.
Colleen Murphy, she's at Princeton. When we perform demanding mental tasks, the brain often has to engage multiple different regions, each of which are specialised at performing specific functions at the same time. And then we exchange information between them. The theory goes that these disparate brain areas link up by synchronising the pattern of nerve firing in both. So what would happen, Ines Violanti wondered, if she artificially enhanced the degree of synchronisation? Would a person's intellect be boosted?
I'm interested in how different regions of the brain talk to each other. So if you have any sort of cognitive process, so if you have to hold information in memory for a couple of seconds, you need different regions of the brain to communicate, to pass information between each other. And people have associated brain waves to those processes.
Are you saying then that you've got a pattern of brain waves in one place and a pattern of brain waves in the other, and the brain matches them up, like syncs them up, so they're almost beating, two drums beating at the same time, and that's how you achieve this sort of coherence between these two brain areas?
Exactly like that. So the idea is that you have millions of neurons in one region of the brain firing up and another in another region, and they reach their sync. And if they do that in a certain beat, you'll be able to pass information.
Now, was that just fanciful thinking? It was a nice model that would explain possibly how things worked, or was there actually objective data supporting that idea?
There's actually both empirical data and using invasive recordings and also other electrophysiologic techniques like EEG, and there's also models supporting the same idea.
So if there's already a reasonable body of evidence supporting this idea, what was it you were seeking to find out about it?
So we wanted to find out whether we can use stimulation techniques to manipulate the way that this information flows, if we can affect behavior. And it will be even more powerful if we can use imaging at the same time. So if we can use brain scans at the same time, and we can understand what we're doing to the brain when we manipulate how this information is flowing in the brain.
So how did you do it?
We use a technique called transcranial alternating current stimulation, which is a form of non-invasive brain stimulation. We combine this with functional magnetic resonance imaging.
Does this mean then that you're literally putting electrical current into people's heads?
That's right. So we did that while people are in the scanner, and we measure their brain activities.
And by using alternating current, does that mean that you can then drive the rate at which neurons are firing? So you'll get that beat synchronization, that's what you're going for, between diverse regions of the brain?
Precisely. So we hope that we are able to manipulate those neurons to oscillate at the frequency that we are imposing.
So what did you ask the subjects to do in the brain scanner then while you did this to them?
We ask participants to perform 2 tasks in the scanner with different levels of difficulty. One of the tasks was quite simple. They just had to press a button that matches the direction of an arrow they saw in the screen. And the other task was a bit more difficult. We show them numbers and we ask them to press a button when the number they saw in the screen matched a specific sequence.
So that latter task, that's a sort of working memory task, isn't it? They've got to hold something in mind, whereas the first task is literally a see, interpret and react task. So you don't have to hold information and share information between different brain areas.
Precisely, yes.
And one would therefore predict that if having synchronous activity in the brain is boosted, then they should improve on the more difficult tasks when you do that.
Exactly, yeah. So that was the prediction, that we will be able to manipulate this process when you actually need to engage different areas of the brain, when the process that you need to execute, to perform, is hard enough that it needs different parts of the brain to talk to each other.
Is that what you saw?
Yes, that's what we saw. The stimulation would only influence performance in the difficult task.
Now, obviously, if there is a reinforcing effect, a beneficial effect of stimulating these different brain regions so they synchronize, logically, if you were to throw a spanner in the works and desynchronize different brain areas, that should impair performance, or at least not help it. Did you try doing that?
Yes, we did. And behaviorally, we didn't see a significant effect of the desynchronous condition.
But is that just because the brain is pretty good at doing what it's doing, and had you stressed the system more, you might have seen an effect?
That might be the case. It might be that if we got to an even harder condition, we would see bigger spread of the effects.
And you had these subjects in a brain scanner while this is going on. You're either stimulating the brain disparate regions at the same time synchronously or asynchronously, so they're offbeat. What do the brain scans show when you do this?
The synchronous stimulation shows an increase in brain activity in the regions that are involved in working memory, whether the asynchronous stimulation shows increase in brain activity in regions that are not involved in working memory.
So that's sort of logical, isn't it? You'd expect if you're just recruiting some brain areas that you're going to get a bit of non-specific brain recruitment, which is not related to the task in hand.
Yes, indeed. But it was quite interesting to actually be able to see those effects and to be able to see that even functional connectivity, a measure of how different areas of the brain are talking to each other, that also changed.
I think I could do with a bit of enhanced cognitive synchronization from time to time, or maybe just a cup of coffee. That was Ines Violanti. She's at Imperial College. And on that note, that is it for this month. You've been listening to the eLife podcast, previous editions of which are also on eLife's website. That's elifesciences.org, or you can subscribe on iTunes. This has been a Naked Scientists production. I'm Chris Smith, and you can find out more about what we do on our website at nakedscientists.com. I'll be back with another look inside eLife next month. Until then, though, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.